I know we mentioned CRISPR-Cas9. We will get to that. Before we do, though, I do want to talk a little bit about enhanced delivery systems. And so we know that improvements in delivery systems can help ensure that the editing machinery reaches the target cells efficiently, thereby kind of reducing the likelihood of off-target effects in non-target cells. And so, Eric, while I have you, I'd like to start with you. Under the umbrella of enhanced delivery systems, what do those improvements look like? What are they? Are they even feasible? What are your thoughts on that?
Yeah, so I think, these can be characterized in ex vivo and in vivo. So ex vivo, typically, you have the cells outside of the body. So you're doing a transfection, ribonuclear protein electroporation, or viral transduction to get your gene editing components inside the cell to make the edit. Generally, these strategies try to achieve a quick peak expression and then quick turnover of those editors to limit the amount of time that they can edit the cells. And it gets a little bit trickier when you're thinking about in vivo, because you have to inject. You have to inject it. Then you have it has to navigate to the cell type of interest incorporate into those cells. So there's obviously a lot more variables there. I think Steve touched on a couple of these, but using serotypes for viral vectors for better tissue specificity is one good strategy. Certain AAVs can cross the blood-brain barrier, for example, for targeting neurons. You can use tissue-specific promoters for viral strategies as well, so that even if it's delivered to an incorrect cell, it wouldn't be able to be expressed unless you because you have that tissue-specific promoter. For lipid nanoparticles, right, you can modify the nanoparticle for better tropism to the liver, for example, for the recent studies, clinical studies with CRISPR. There's some new techniques for targeting strategies, including cell-specific aptamers, selective organ targeting, or SORT, for trying to target the nanoparticles to specific organ systems as well. And even some lipid nanoparticles have been much more common and come onto the scene a lot more with COVID targeting strategies as well. And then peptide-based approaches have also been considered as well. So there's some interesting research in this area as well for coupling your targeting peptide to the CRISPR components using a cationic complexing peptide to an anionic CRISPR component. And these strategies might be more prone to degradation as they're a bit more exposed than lipid nanoparticles, but an interesting area to consider as well.